Next up in our series on biomolecules, we have lipids. And as you can tell from this meme, they don't mix well with water. So that is going to be a foundational property of lipids. Lipids are nonpolar. That means they are hydrophobic, which means they do not mix with water. And lipids are a little different than some of the other biomolecules. The other biomolecules have a lot of similarity in structure, so you can pinpoint a specific monomer and you can kind of put those monomers together to make the polymers. But lipids are more classified by the fact that they don't mix with water. So that is a fundamental property of all lipids. They are all nonpolar, and that is going to give you some differences in structure and then also some differences in function of the different lipids. And lipids and a lot of these biomolecules are kind of almost like Legos, where you can piece together different pieces or different monomers to add different functionality. So the structure and function, like with any biomolecule, come from the way you assemble those components. And if you're wondering why I'm just pointing that out for lipids, it's because it's directly stated in the course and exam description that tells me what to teach y'all. But even though there isn't technically a monomer for all lipids, a lot of times what you'll see in a lot of these comparisons between biomolecules is fatty acids listed as the monomer for lipids. So let's talk a little more about fatty acids, because one of the two major categories of lipids we're going to talk about today are fatty acids. And you can call fatty acids either saturated or unsaturated. So saturated fatty acids only have single bonds between their carbon atoms, like you can see in this picture. And then unsaturated fatty acids are going to contain at least one double bond between the carbon atoms. And the importance of that is that makes the carbon chain bend, like what you see in this kind of fatty acid simplified diagram. And we say that those are kinked or bent fatty acids. So the single bonds, they're capable of some rotation, and the single bonded fatty acid chains tend to form these straight lines. Whereas the unsaturated fatty acids have these double bonds that can't rotate and cause the fatty acid chain to bend. I do also have trans fatty acids on here, but we're not going to talk about them much because those are pretty much artificial, that are made by humans. They're not really found much in nature, and if at all, and they're not healthy for you. So let's talk about why this unsaturated versus saturated bit is important. It has to do a little bit with the fluidity of these lipids. So if you have no double bonds, not double acids. If you have no double bonds in your fatty acid chain, then that's a saturated fat, and these guys tend to pack closer together. So they are solid at room temperature, and you have to add more heat to them to melt them, which is important because if you are, say, an organism living in higher temperatures, you might want to have more saturated fatty acids in some of your molecules just so they won't liquefy as easily. If you have less double bonds, then you're going to have, but if you do have some double bonds, like if you just have one double bond, that's a monounsaturated fat. And notice these are liquid at room temperature, and they have a lower melting point than saturated fats. If you have a unsaturated fat, it has multiple double bonds. It is liquid at room temperature, but it also has the lowest melting point of all. So the more unsaturated a lipid is, the more double bonds it has, the more liquid, more fluid it's going to be at room temperature. And like I said, we're not even going to care about trans fats because those are not found in nature. And that's going to become important in a little bit. But for now, let's talk about some of these different types of lipids. So the one we've already seen kind of some examples of, and that is going to contain fatty acids, are fats. The more common form of which we'll see in a lot of biology textbooks and the biology course are triglycerides, or triacylglycerides as they're sometimes called. They are made of glycerol with 3 fatty acid chains, and their major function is going to be energy storage. In animals, these are stored in fat cells, which are sometimes called adipocytes. Adipo literally means fat. Cyte means cell. And an important function that they perform in mammals is to help insulate and help trap heat to keep mammals warm. But like I said, they also are a very efficient form of energy storage in that they can store more energy in a smaller space than carbohydrates. So they're more used for long-term energy storage, more so than carbohydrates, because that would require you to set aside more space in the body to store the same amount of energy. Steroids are the other general category of lipids that we're going to talk about. Steroids are not like what you think, what you hear about on, online or social media. They're not all like bodybuilding chemicals. You actually have a lot of natural steroids that act as a variety of hormones inside the body. And so they play a role in a lot of different functions. You have the ones that are involved in gender determination, as well as when humans go through puberty and animals go through puberty. So aldosterone and testosterone, those are going to be some of the androgens or male sex hormones. And then estradiol and progesterone, those are two of the estrogen family or female sex hormones. Then you have some of the steroids that perform other functions. Cortisol is a really big one, and it is a stress hormone. So it is released in your body when you're facing physical stressors or emotional stress. So that's going to play a role in mostly like homeostasis maintenance. And then you also have some roles in energy metabolism. You do not need to know the differences in the structures of these. The major thing to know is, one, a lot of these are synthesized from cholesterol, which is a very specific steroid. And then also the fact that these are non-polar makes them fat soluble, which we're going to talk about later when we talk about hormones and how they work to act or cause responding actions in cells in Unit 3. So let's talk a little more about cholesterol. One thing I want to point out, cholesterol has a large nonpolar tail, and then this region is also nonpolar, but it does also have a polar head. And that, when we talk about cell membranes later, is going to come to play because cholesterol is often found in cell membranes. It's very important to them. It helps to stabilize them and make sure that even at high temperatures or low temperatures, that these cell membranes aren't too fluid or too rigid. And we'll talk about that in more detail when we talk in detail about the structure of cell membranes. Speaking of cell membranes, the most essential molecule in them are phospholipids. So phospholipids are very special molecules. They have both polar ends and they have non-polar ends. So they have both hydrophilic areas, which are attracted to water, and hydrophobic areas, which are repelled from water. And this makes them form a very effective barrier. And really quickly, they're going to become a huge role in cell membranes. We'll talk about them in more detail later. Here's just a very brief summary slide with some of the functions, where they're found in cells. Notice I have an asterisk next to the monomer as fatty acids because it's not the monomer of, say, for example, cholesterol or the steroids. And keeping in with the theme, since I had the sweet cats on the other slide, here's my fat cat. You can't see his fat very well here, and I'm not going to point it out because I'm trying not to embarrass him because he will bite me more.